We talked about chromatin modifications. Certain certain ones promote certain promoters are demethylated, and methylation of lysine 4 and histone 3 are trimethylation. Then there's DNA level modification and nucleosome level modification. Transcription factors are coming from mRNA in the egg. Maternally present transcription factors bind to the promoter chromatin modification. And then we talked about the veg-T protein transcription factor.
What it does?
It makes the endomer.
It tells the vegetable cell to become endoderm and cell above them to become mesoderm, right? Okay, now we are talking about gastrulation. Okay, so what happens during the process? So gastrulation starts from blastula, blastula changes to gastrula, right? And we also talk about there's something happened what we call mid-blastula transition, where a few new genes start expressing. So there are three things that need to happen during the gastrulation. Number one thing is that the blastula, because gastrulation starts in the blastula, right? So what this does, blastula brings in the embryo. What's it bring in? Those cells need to become mesoderm and endoderm. So blastula need to bring those cells inside from outside. Number one. Number two, it should surround the embryo with the cells. And those cells will become ectoderm. So these things they have to do. And most important thing is, it need to place mesoderm between ectoderm and endoderm. So, what ultimately we are going to get? Ultimately we are going to get an embryo. That embryo will have 3 layers, okay? So what we are going to have is, we are going to have number 1, so we will have an embryo that will have 3 layers of cells, okay? Now these are called, these layers are called germinal layers. So we'll have 3 germinal layers, okay? So outer germinal layers will be the cells, what you call ectoderm. And the ectoderm are going to give you skin, going to give you nervous system, and neural crest. Okay. And then you will have these cells, which are in the center, these are called mesoderm. And mesoderm is going to give you skeleton, muscle, circulatory system, excretory system, and your most of the reproductive system. So that is all coming from the mesoderm. Whereas your endoderm is going to give you your digestive system. Digestive system and respiratory system, and thyroid also come from there. So this is all coming from the endoderm. Now this cavity there is, this is called archenterome. So this cavity is there. This is called archenterome. Archenterome. Okay, now, so this is the gastrula we are going to get, okay? So there are 3 germinal layers, and this is called triploblastic. So the gastrula with, so we are all triploblastic animals because we originate from 3 germinal layers, okay? Now, here is important thing when we talked earlier. Now, when this is, any question?
What? Which one is the green?
Green one is this.
Yeah.
That is
the
endoderm.
endoderm.
Oh, sorry, I should have shown it, did not do it. That is endoderm. Here it is. Okay. So now, if we look here, what we are observing here is, now this is pretty early embryonic stage, right? But if you see here, no, all the 3 germinal layers, they are giving different type of cells, right? So that means even at this stage, they are not pluripotent. So pluripotency means cell can give every cell type of the body. So at this time, they cannot. Because these layers have specified that what kind of cells these different layers are going to give. So we all are originated from 3 germinal layers of the cells. So we are all triploblastic. There are some diploblastic organisms also, but those are lower organisms, right? So we are all triploblastic. So what we are saying? We are saying the gastrulation is the process by which the blastula need to bring the cells from outside to inside. So it need to bring the cells which are going to give endoderm and mesoderm. Then it need to make sure that the cells can cover the whole embryo to form the ectoderm. And it also makes sure that mesoderm is placed in the center of, in the middle of ectoderm and endoderm. And we are saying these 3 germinal layers, for example, ectoderm is going to give you skin and nervous system and nerve crest. Mesoderm is going to give you a variety of skeletal, muscle, circulatory system, excretory, and most of the reproductive system. Whereas your endoderm is going to give you digestive and respiratory system, and in the center we have described what we call archenterome. So far it is okay? Okay, now. So how this process occurs, that's what we want to talk about. Okay, so for that purpose, let me show you first this thing and then we'll go further. Okay, so this is what
When we look at
happens.
a cross-section of an embryo of the frog xenopus, we can see that at this point it is a ball of cells with a fluid-filled cavity. The cavity is the blastocele, and the embryo is currently in the blastula stage of development. A blastula contains large yolk-filled cells at the vegetal pole and smaller cells at the animal pole. The three colors represent the three tissue layers that become defined early in embryogenesis. Yellow indicates endoderm, red indicates mesoderm, and blue indicates ectoderm. At the beginning of gastrulation, a few surface cells, called bottle cells, move into the interior of the embryo, followed by other surface cells. We can track the movement of cells into the embryo if we add dye to a few surface cells. The movement of cells into the embryo creates a lip, called the dorsal lip, over which sheets of cells continue to move inside. At the same time, the ectoderm extends around the embryo surface in a process called epiboly. As gastrulation proceeds, a cavity called the arc enteron forms while the blastocele progressively shrinks. The arc enteron is the primitive gut and is completely surrounded by endodermal tissue. The endoderm at the roof of the cavity originated from the outside of the embryo. The cavity is continuous with the outside via the blastopore, which eventually becomes the anus of the animal. As the ectoderm extends around the embryo, another set of bottle cells forms. These cells migrate into the embryo, and other surface cells follow them, creating the ventral lip of the blastopore. By the end of gastrulation, the ectoderm has surrounded the embryo. Endoderm lines the inside, and mesoderm lies between the two. Additionally, the fates of specific regions have become determined.
Okay,
If we look at a cross-section of an embryo of the frog xenopus, around the embryo, another set of bottle cells form.
okay. So let's talk about how this is kind of, we saw what is happening from here. The cells have started moving in, right? So how that process is happening, what is happening, we're going to talk about each step there. Now, we're talking about gastrulation, okay? So what happened that in the blastula, so blastula consists of numerous cells, right? Now the position where each cell is, that is determined by the cleavage, right? Because the cell divided, and we said top are all micromeres, bottom are megamere, the smaller blastomere, bigger blastomere, then a blastocele is formed. What happened there? Just the cell division, right? But during gastrulation, what is happening? These movements are bringing cells from outside to inside, making new neighbors, right? Making new position. So all those new neighbors, new position are going to determine what these cells are going to be. So then what kind of these movements are taking place during this type of migration of the cells, right? So what we are saying, so we are talking about the cell, so blastula changed to gastrula. But gastrula require the movements of these cells. So what types of movements are taking place? First we will need to know those, okay? So we are saying the types of cellular movements. What types of cellular movements are taking place during this process of gastrulation, how the cell is moving. Now the first is what we call is invagination. Invagination. So what is invagination? Invagination is essentially infolding of the sheaths of the cells. For example, if you have this embryo like this, in this embryo, right here, an enfolding will take place, kind of a finger like these are all the cells, they are kind of enfolding inside. Okay? So that is called invagination. So they kind of like fold inside. Making sense? Kind of like getting it, invagination. Then you have the other movement that is called involution. So involution. So what is happening there? That is the inward movement of expanding cells along the surface layer of other cells. What does it mean? Look here again. So if you start looking from here,
Few layers that become defined early. It's endoderm. Ending of gastrulation. Cells move into the interior of the embryo, followed by other surface cells. We can track the movement of cells into the embryo
Now
if
look
we
here.
add dye to a few surface cells.
so what is happening? These cells are dividing, growing, and they are moving inside along the surface of these cells. Making sense? So that process is called involution. So these cells are dividing, growing number of cells, and they are moving inside along the surface of these cells. That is called involution. Okay, so essentially these cells were initially outside, they were not inside. But they started dividing, growing inside and covering it. That is called involution. Now, then there's another thing what we call ingression. Ingression. Now what can happen that, for example, this is the embryo. From here, this is, for example, so these are all the other cells here. From here, a single cell can actually break. So a single cell from here break and get in. A single cell. That is called ingression. Okay? A single cell break and gets in. Now, then there's another process what we call delamination. Delamination. What is delamination? Delamination is actually splitting of one. So here in ingression we just talk about one cell, right? When we talk about delamination, what we are saying, splitting of one cell layer in two or more parallel sheets. What does it mean? Let me explain here. So, for example, you have these cells there. These cells are there. These cells divide in a way that they divide from the center like this, divide from the center like this, from like this, from like this. And when they divide from the center, what happened? That initially you had the one cell layer, now you have a two cell layer right there. Making sense? Right? So that is what is happening. Splitting. Initially it was one cell layer. That one cell layer split and make the two cell layers. Making sense here? So that is called delamination. And then there is a process of another type of movement, and that movement is called epipoly. So 1, 2, 3, 4, and 5 is epipoly. Now epipoly is essentially movement of epithelial cells to cover the whole embryo. To cover the whole embryo. Now what is that? So for example, this is our embryo. Right here, so these are the cells of your vegetal pole, this is your animal pole, right? So what happened? These cells which are there, these are going to be epithelial cells, these are ectodermal cells. What these cells do, these cells start migrating down like this, and they essentially cover the whole embryo. That is called epipoly. So we are saying that gastrulation involves 5 different type of movement of the cells. Okay. Now what are those movements? We are saying that first movement can be invagination. Invagination is essentially infolding of the sheet of the cells. Okay. So you have these cells have kind of infolded inside. Then the second movement can be involution. Involution is inward movement of, I should have write outer layer, because these cells were from the outer layer, right? So inward movement of expanding outer layer of cells inside the embryo, developing embryo, along the surface of the cells. That is involution. So outer layer of cell, they started moving inside, and their outer layer, they are expanding, and they are moving along the surface layer of the other cells. That is making sense here? Now, then we are saying that next movement can be ingression. Ingression can be out of these cells, one cell split and get inside the embryo. That is ingression. Then we are saying splitting. Splitting is essentially, we are saying that the whole cell layer split into 2 or more layers. And that is happening because these cells divide and they create another layer beneath those cells. And then we are saying that finally there is a movement called epipoly. And epipoly, where the outer layer of cell, here in case the epithelial cells, they start dividing and they move around the embryo to cover the whole embryo. That is called epipoly. Making sense so far? Any doubt? Okay, so talk to each other for a second, then we'll go further.
ingression, one cell splits and releases one B cell into that. Delamination and splitting of one cell layer into two or more parallel sheets. Fully movement of epithelial cells take over the whole embryo. The animal hemisphere. This is from, like, the ones that cover it are from the animal hemisphere.
Just looking at one side, like
Right.
two or more parallel sheets. And then epithelially. That's not from the animal pool.
Okay, any doubt here? You're okay, Mason?
Yeah.
Okay, now. So, we are observing two things here. Number one, these cells are going in, then this ectoderm is covering. Now we are basically trying to talk about how these things are happening. Okay. So first thing we are going to talk about the epibole of prospective ectoderm. And first, let's talk about what is epidermis. Okay. So epidermis is the outer layer of skin that is derived from ectoderm and covers the whole embryo. Right? That's what we are covered with the skin throughout. So that is, so it means cover the whole embryo. That means ectoderm from vegetal hemisphere is going to cover epidermis from animal hemisphere, sorry. Going to cover vegetal hemisphere. Right? So what is happening? So we are saying that, let's see, this is our we're trying to draw at the blastula stage what is there. So we are saying this part, these are all cells here. So this is your vegetal pole. This is your animal pole. Okay? And what is happening here? Here you have all those cells right there. So these cells are there. This is going to undergo the process of epibole, and ultimately we are going to have these whole cells so this ectoderm is going to cover the whole embryo. Okay? So this will be the outcome of this whole process. Now, why and how this happened? There are two mechanisms which help the epibole to take place. Number one is increased cell division. Increased cell division. So what is happening there, for example, in this area, in the top area, right here, what is going to happen, that these cells which are there, these cells will start dividing. They will start dividing. So you will start making more and more cells from here. These cells will start dividing, okay? So, increase the number of cells, that's the first thing which is going to happen. And then, what is going to happen is, what we are saying, that's the first mechanism. The second will be integration of the cells of the deeper layers into the surface layers. So the cells which are in the deeper layers, they will integrate into the cells of the surface layer. And that is going to happen through radial intercalation. And what is that? I'm going to explain. So, multiple things are going to happen. The cell number has increased. So here, for example, initially it was maybe like this, right? And then the cell division has taken place. You have more and more and more and more and more cells there. The cell have increased. So because you need more cells to cover the whole embryo. Now, what is going to happen? Two things are happening. Now, let me draw these cells a little bit bigger. So these are your surface layer cells, okay? And under the surface layer, you have these cells are there. These cells are deep layer cells. I'm writing here as deep layer, okay? So what happened that the deep layer of cells deep layer cell, DL, sends protrusions into the surface layers. What is that? For example, this cell is there, this cell is there. What they do, they change the shape in a way, let me draw it here. So the cell which are here, they are like this, but the cell here, they change the shape, kind of like they go and send the protrusion like this, okay? So these protrusions are sent in them. Same way, this is the other cell right here, and the cell here, which was there, that also sent a protrusion like this. So they're kind of like making a protrusion right here from here, sending a protrusion right here, sending a protrusion right here. Okay? And what they do, then they physically move from here, they physically move from here to here. So, send protrusion into the surface layer, that's the first thing. And second, they physically intercalate between the cells of upper layers. So then it physically move up, and you will have now like this. But now you will have, so you have 3 here, 1, 2, 3, 4, 5, 6. You have 6 cells there instead of 3, right? So then these cells which are on the surface cell, they actually expand. So these surface cells, the surface layer cells, they expand. So this cell will expand like this, make bigger itself. This cell will also expand, make it bigger. And this cell will also expand. So these cells essentially expand. Making sense? So what is happening? Two things are happening here. So if you had the cells like this, right? They have increased the number. If they have increased the number, that means more number of cells. So think about, in this layer, you had only this many, you initially started with 3 cells, and this is your embryo, right? Now you become 20 cells. Now what you're doing? You're entering into the other layer, and you're expanding also. So in this way, when you're expanding 3 layer, becoming 1 layer, so you're kind of expanding it, making sense or not? Right? Do you understand what I'm trying to say? Yes or no? I didn't get it.
So the deep layer is kind of like moving up to expand the.
Yeah, deep layer is moving in, and then these are also expanding. So what is happening, if this was covering only this much area, right? So then now these cells are covering this much area, making sense, the same number of cells. So in this way, they can cover the whole embryo by this process. So then, what is the reason why these cells move from here to here? Means what is the reason? So what happened that these surface layer cells, the surface layer cells, they secrete a chemical, and that chemical is called C3A. So they secrete C3A. And that C3A can act as a chemoattractant molecule. So if that has to act as a chemoattractant molecule so that these deep layer cells can move up, then that means these cells should have a receptor. So these surface layer cells, they all express the C3A receptors. So what is happening? We are saying that epibole is a process by which these ectodermal cells are going to cover the whole embryo to make epidermis, the part of the skin, to cover the whole embryo. And we are saying that process happens because of the 2 main mechanistic events which are taking place. Number 1 is the increase in the number of cells. So the cells which are right here, they start dividing, they increase the number of cells. And when they increase the number of cells, that means the number of cells have gone way high than it was earlier. Then the second thing happened, they do a process what we call radial intercalation. And radial intercalation is the process where these deep layer cells, they essentially send the projections in the cell above them and then physically move between the spaces of those 2 cells called physical intercalation. And we are saying that, and then the surface layer cell, they kind of expand, and in this way, the surface, the ectoderm layer starts expanding. And we are saying that process takes place because these surface layer cells, they secrete a chemoattracting factor C3A that attract the deeper layer cells toward it, and that happens because these deeper layer cells, they express a receptor called C3A receptor. So that leads to intercalation, and that intercalation is followed by the expansion. In this way, the epibole takes place. That is making sense? So talk to each other for a minute, then we'll go further.
The epidermis is the outer layer of skin surrounding the epiderm and covers embryo. It covers making the animal center and covers the whole thing through the process of epibody. Two mechanisms are increased coloration and integration of cells with deeper layers breaking
Yeah.
the surface layers, there's like interpolation, and then a bunch of pictures where there's some protrusions out, and then they physically interpolate with upper layers. And then the upper layers also expand. And this is because the surface layer secretes urine.
What is not clear? You good? Okay. You guys are good here? Okay, now. So, this cell, they are going to cover the whole thing. Now we are saying that, if you see here, we are saying at this point, before the movement, these cells called bottle cell are there, okay? Before the involution start. So what is happening there? So the cell movements of gastrulation, that is not happening everywhere. That is happening at a special place. And that place starts at future dorsal side of the embryo. In the gray crescent, we know that gray crescent opposite to the point of sperm entry. So from that is the place where the movement start, okay, and where it happens. So this is the your embryo, animal, vegetal, right? Right here is the equator. This is the equator. And these cells right here, which are right at the equator, these cells, what we call them is a marginal zone. Right here, these things are going to happen. So what happens? If I kind of like basically make it a little bit bigger, look at the cells. So what I see here, I will see these cells on the animal hemisphere are smaller, cells in the vegetal hemisphere are big. Okay? So what is happening there? So this will be like this. Okay? Now, so these are the cells at the marginal zone, right here. This is happening right here. This is the area. Okay? So that means, so out of these cells, say some of these cells right here, it's the marginal zone. What they are doing, the cells at marginal zone change their shape dramatically. They change their shape dramatically. And what they do, that the main body, so this is the cell, so main body of the cells is displaced. Okay? So what happened? The main body of the cells is displaced. So these cells are there. Now see what is happening from here to here. This is the second step. that these cells which were there, their cell body is displaced in a way that they are like this, form structure like this, form a structure like this. So what that is leading to, maybe I should have drawn it a little bit further away. Let me draw it here. So main body is displaced in a way that form a kind of a bottle-like shape, okay? And they are attached to rest of the cells by these thin cytoplasmic connections. So what happened? Then these cells which were there up, they're kind of like the cell which was here, they're attached like this. And then you have the other bigger cell which was right there. They're attached like this. Now what has happened? The cells which were here in the marginal zone, they have changed their shape. They have become kind of bottle-like cells. Okay. Now, what it has done, it has caused the cells above here and cell below here to come in contact with each other. Making sense what I'm saying? So these cells, this cell and this cell, they are far away from each other. Okay? Now the cells right here, this is happening just, actually you should not have drawn it here. This is happening just beneath the equator. Right here, this process is happening right here. So this process is happening right here. Okay? So these cells which are here, they become the bottle cell. And then the cells which are here, they are coming with contact with these cells because these cells have changed the shape. Making sense so far? Everyone is okay? So cells of animal pole, animal hemisphere, has come in contact with cells of vegetal hemisphere. So because the cells at the marginal zone, they change their shape in a way that they form a bottle shape, and they are connecting to the rest of the cell just by a small cytoplasmic connection. That caused the cells in animal pole to come in contact with the cell of the vegetal pole. Now, then a constriction is formed. You can see a constriction is formed here. That constriction which is formed here, this is called blastopore. If you think about, if this is the blastula right here, now this blastula has formed a pore-like structure which is there, right? So this is called blastopore. And now if I look here, this cell is there, and these two cells which were far away have formed this connection right here, and you have these bottle cells are being formed. Making sense here? So these are your bottle cells. And then this upper cell has made connection with this cell. Making sense or no? So you have now a blastopore lip here, a blastopore. And the cell which are here, so this portion, right, this cell which is here, this is called the cell of dorsal blastopore lip. Okay. So once these cells connect with each other, the vegetal cell connect with the cell of animal hemisphere. these cells, they start moving in. They cell from here, they start moving in. The involution start. Therefore, once the cells of animal hemisphere comes in contact with cell of vegetal hemisphere at equator, and that is the place what we call gray crescent. Immediately what happened? Involution starts. Okay. Now what is happening? These cells are involuting, migrating in. Okay. So this area where the involution is taking place, that is called involuting marginal zone, also called IMZ. Now, if you are saying, if you go here back to that picture, so we are saying that first bottle cells are formed, okay? Once these bottle cells are formed,
Interior of the embryo, followed by other surface cells. We can track the movement of cells into the embryo if we add dye to a
then these cells moving in, right? So these cells which are here, they are moving here, dividing and turning in. That means what is happening, if you look at more carefully,
few surface cells.
so we are saying these cells are moving. So every time the type of cell changes, because the cell here has gone in, then cell forward come, goes in, the cell comes, cell goes in. The type of cell at IMG, or involuting marginal zone, changes as the cell are moving in. Making sense or no?
Can you repeat that one more time?
So, in the beginning, this cell was here, right? This was a different type of cell, then this type of cell, then this type of cell. So, at the IMZ, the type of cell is continuously changing, because every time a new type of cell is getting in. Making sense? Because this cell first enter, then this cell enter, then this cell enter, then this cell enter, then this cell enter. So what we are saying? We are saying the cell movement of gastrulation, they are initiated at the future dorsal side of the embryo in the gray crescent area directly opposite to the sperm entry. And we are saying that happens because the cell which are here in the marginal zone just beneath the equator, they change the shape. And they change the shape. When they change the shape, they are displaced inside the embryo. They were outside, they are displaced inside the embryo, right? And when they are displaced inside the embryo, right here,
Cells called bottle cells.
yeah. So you can see they are displaced inside the embryo. So they form a bottle shape, and they are connecting to rest of the cell by thin cytoplasmic connections. What that does, that bring the cells of the animal hemisphere in contact with the cells of the vegetal hemisphere. And that create a kind of like this, what we call a blastopore is formed. And then these dorsal cells, which are right here, which are now called dorsal blastopore lip, from here, these cells which were on the surface, they actually start migrating or involuting inside. Okay? And we are saying this area from this involution is taking place, that is called involuting marginal zone. And the cells of the involuting marginal zones, they are keep on changing as these cells are migrating inside. Now, here is important thing. Once the involution process starts, once they start migrating in, at that time, even if you remove the bottle cell, the involution will keep on going. because the purpose of bottle cell formation was to bring in contact, for example, this cell with this cell, right? Once they start are in touch with each other, the involution starts, then their role is done. Making sense? Anything which is not clear? Okay, so talk to each other for a second, then we'll go further.
Future dorsal. Migrate percent, which is
Hey, Dr. Vore, when you said the cells change as they move up the surface of the
What
layer,
changed?
so these cells, as they come and move up like that, you said that they were changing?
Yeah, so changing means this cell, this cell. Now cell number 1, cell number 2, cell number 3. First cell number 3 gets in.
Okay.
Which makes the black.
So the next person here is not cell number 3, that is number 2.
Cells that were referred to
So for
as
example, first you enter, then Rachel entered, so every time the person here is not the same.
Cell of the dorsal
That's what I
capsule
got.
Okay, okay,
causes
I got it.
evolution to cell.
Thanks.
Once cell of the animal hemisphere comes in contact with the cell hemisphere, they create evolution.
Any confusion here? So you understand the concept of the cell changing, he had that thing.
Yeah.
Right, so every time it will not be the same cell, right? For example, first you enter, then she is going to enter, so it's not the same, right? Any other thing here?
Do you mean like the same like cell in general, or do you mean the same type of cell?
Oh, they will be different, we'll talk about, every cell is different. I mean, they look very similar from outside, but every cell is going to give you different thing,
Yeah.
right? We said, they'll be coming mesoderm, so mesoderm gives so many things, some muscles, some bones, so.
So I was kind of confused about the marginal zone.
Mhm.
So is it more of the vegetals?
They-
part of
No,
the
these
cells,
are
or
these
is
are
it
these are
not
kind
the
of
same?
like more of the vegetal area cells.
Okay.
Yes, there's a more of the vegetal area cells. No, yeah, vegetal is not. Yeah, more of the vegetal area. Yes. So anything else which is not clear? Okay, so what happens?
Dr. Vora, we only have like 2 minutes left.
We are done.
We have 2 minutes left.
We have just 2 minutes left. Okay.
Move into the interior of the embryo, followed by other surface
Okay,
cells.
then we'll.
We can track the movement of cells.
We
into
can
the
watch it
embryo
once more
if
and
we
then
add
show
time
that this
to a few
picture.
surface
I'm not going to
cells.
draw this thing, but I will explain you how
The
this
movement of
thing.
cells into the embryo creates a lip called the dorsal lip, over which sheets of cells continue to move inside.
So here is one more thing happening. As these cells are moving in, they're entering in the blastocele, right? So when they're entering in the blastocele, you can see they are making another cavity here. So blastocele will be gone and another cavity will be formed. And that will be primitive cut. I draw there archenteron, okay? And we'll keep on talking about this. So we will meet tomorrow in the lab and we'll go from there.